This is The Dime, a 10-minute dive into the cannabis and hemp industry through trends, insights, predictions, and tangents.
What's up, guys? Welcome back to another episode of The Dime. As always, I've got my right-hand man, Kellan Finney, here with me. And today we've got a very special guest, Jordan Zager, CEO and co-founder of Dewey Scientific. Jordan, thanks for taking the time. How you doing today?
Hey, yeah. Thanks for having me on. I'm doing good. How are you two doing?
Awesome. It's a beautiful day here in New York at the end of March, and we're excited to kick it off.
Same. It's pretty warm out here in Colorado, so
I'm jealous. It's about 42 degrees and sleeting here in Washington right now. It's a normal spring.
I think before we get started, I think it'd be great if you could share with our listeners a little bit about you and what Dewey brings to the space.
So a little bit about Dewey. We like to say that we're bringing incredible science to the cannabis industry. And when we're talking about incredible science, we're talking on the plant genetics and biochemistry side. Because, that's really what cannabis is in the end, is it's a plant that has very unique genetics and biochemistry. So we really try to apply modern science techniques to understand that and then how to leverage that into commercial cultivation and plant breeding. My background, I have a PhD in plant biochemistry and was one of the early people to work with cannabis genomics within academia. And I've discovered a few terpene genes, some synthases in the terpene family.
How did you get started in the cannabis industry?
Yeah, so I'm going to go pretty far back here to my days in college. I worked in a plant biochemistry lab, and as a college kid, I was smoking weed all the time. I was like, wait, I'm learning some really cool stuff about plant biochemistry. I happen to love this plant. Let's make this my career. At the time, there was medical in some states, and then recreational just started in Washington and in Colorado. So when I applied to grad schools, I only looked for states where I could work with cannabis, maybe at home, not necessarily in the lab. That brought me to Washington. I found a lab whose main focus was the biochemistry of glandular trichomes and terpene production within secretory cells. So, I knew enough about plant physiology at the time to, trichomes, terpenes, sounds like cannabis. It's going to be pretty close to working with cannabis. So I joined that lab, and then eventually I got to work with cannabis, part of my PhD project. And it was about that time that project started. I decided, let's try to apply this into the cannabis space. And it's not like at the time there were a bunch of, large companies like there are today, at least in Canada, where they have pretty in-depth research teams. And so in the time since we've founded Dewey, more and more companies have incorporated this in. But that was my path to the cannabis industry. Just a love for biochemistry and a love for cannabis.
Perfect combination. So before we hop into the questions, take us back to Dewey. How did that start? Was that another cannabis-induced idea? And kind of take us through that.
Yeah, so Dewey Scientific, it was founded by me and a plant breeder when we started out as friends, umpiring baseball. Actually, we were baseball umpires. And after a game, I think we went and smoked some weed, and it turned out he was a plant breeder. I was a biochemist, and so we thought, hey, let's try to apply what we know to cannabis. So I think that was in 2015, 2016. We set up a little makeshift lab in my house. We bought some laminar flow hood on eBay, started doing some tissue culture in my house. And that's how it really started. And then, fast forward a few years, we start pitching to investors and we get funded. So really what we do is we work with other companies' breeding programs, and we sort of supply them with, the scientific expertise as it relates to genomics, genetics, biochemistry to improve their breeding program. And so we work with a few folks in the hemp space. We work with a couple of different farms in the recreational space, farm down in California, a couple of farms in Washington. And then, of course, we have our own recreational cannabis brand, the Dewey Cannabis Company, that's in Washington. And that's it in its whole. We do work with a couple of international companies. We've worked with some folks in Costa Rica. They recently just changed their laws. They've got a CBD market now. So we helped with the first import hemp seed to Costa Rica. Those seeds came through the Dewey lab at one point. So that's really what we do.
Nice. It's the classic tech garage story turned for the cannabis industry. So it's awesome to hear the evolution of it for our space. So yeah, let's kind of dive. Yeah, sorry, continue.
I was just going to say the first, right, to do tissue culture, you need an autoclave. So just high pressure, high temperature. So we bought a Shopco down the street, was going out of business. So we picked up a $20 pressure cooker and That was how we made our first batch of media. $20 pressure cooker in my garage.
Were you living at home at the time?
No, this was while I was in grad school.
You can imagine what your mom would say if she opened it. She's like, what the hell is going on here? So let's dive into some of these things. On the website, it says better crop decision. In its simplistic version, how does Dewey do that?
So we are big fans of collecting data on your crop. If you're going to want to improve something, you need to know what is going on in the first place. So that's sort of our approach there is capturing as much data, whether it's plant performance data in the field or if it's data related to the genetic makeup of the plant. We take a full view of that to then make better breeding decisions.
So let's talk about those better breeding decisions. Are there ones you can specify, ones that are common in the space?
I think with the increase, if we back up to when hemp became legal, with the 2018 Farm Bill, since then there's been a lot of progress by many companies in improving the genetics that are out there. Right, I think the first couple of years everything was like a cherry wine background. Now there's maybe 8, a few dozen of solid backgrounds that are out there. So I'm going down a rabbit hole here. Genetic uniformity was a big problem with the 2018 Farm Bill. Like crops were very uneven. A lot of crops were hot. I mean, that's still a big problem in the hemp space. You guys know that. So it's a matter of, right, if you're just breeding for low CBD, low THC abundance in CBD backgrounds, right? You want to have the tools that can speed up your breeding decisions and your timeline to getting to what you achieve. I don't think that answers your question.
When I was thinking about this, and correct me if I was wrong, have you heard of the myostatin gene that was recognized? It's called the mighty mouse gene, right? And so when I was actually thinking of what Dewey does, I was thinking about this kind of work that's being done like by biohackers, I guess, is the best analogy I could think of. And so the presence of this gene, or the absence of it, causes muscles to increase dramatically, right? And so when I was thinking about Dewey, I was thinking that's exactly the type of work they do in terms of analyzing the plant to say, okay, like I see that, say, this specific strain has massive trichomes, right? And then all of a sudden it's testing higher, but what is the exact reason that it has those larger trichomes? And why is this strain yielding better when I extract it versus this strain isn't, right? And so is that a good representation in terms of Dewey is going to come in and help me learn exactly genetically what's going on from a expression standpoint, and then I can try to replicate that in my breeding program? Is that something that's similar?
Absolutely. Yes. So, right. You just have to have your trait of interest. And when I say, you need the data on it, you want to know, let's go back to your this plant is extracting extremely well. So what we would do is to really learn the genetics behind that, you have to set up a breeding scheme. You want to have something that you want to set up what's called a segregating population. You want to have one parent that has the trait. You want to have another parent that doesn't have the trait. You cross those, and then you look at the progeny, the children of that cross. And if some of them have the trait you want and some of them don't, you have what's called a segregating population. And that's sort of the best way to go about understanding the genetics behind a specific trait of interest, whether it's powdery mildew resistance or it's extraction yield. That's sort of the approach we take. So specific examples of what we've done in the past are, one is powdery mildew resistance. We've developed molecular markers for that. So once we have these molecular markers in hand, it speeds up our breeding process. So rather than, we've got this Skittle mints and we've got this lemon meringue that we want to cross, and this lemon meringue super susceptible to powdery mildew, but this Skittle mints is not. We can make the cross instead of, going through the whole timeline of phenotyping all those progeny. You then just have a quick genetic test at the time they sprout their first leaves to identify what is going to be powdery mildew resistant. So we've done that with a few different traits.
That's awesome.
Yeah.
How long does that take? I know that's ballpark, and that's a pretty specific request, but just ballparking it.
Yeah. So it's really a matter of, if you know the plants you're working with, we're talking about a 13, 14 week timeline.
And if you don't,
that's not too bad.
And if you don't, well, then you got to start collecting data. So that's sort of where we come in with the cultivators, and just provide them the scientific assistance to get what they want, right? Because most operators in the space right now, they know of what's in their genetic library, what does well for certain things, whether it's terpene accumulation, whether it's, purple flowers or what have you, or, resistance to different, abiotic factors or biotic factors, such as right now at Dewey, we're trying to tackle aphid resistance and at least develop some markers for aphid resistance. And so of all the different varieties that our recreational grow is growing, we do know some are incredibly susceptible. And we know that some, I don't want to say that they're fully resistant, but we're almost never going to find an aphid on them. So that's sort of the next trait that we're trying to capture. And so with this, because we haven't really categorized our plants for this specific trait, the trait of aphid resistance, it takes a little bit of time there. So maybe 3 or 4 flower cycles. So that is sort of a long timeline if there's 5 flower cycles in a year. But a year to 18 months, I think, is a good timeline.
And what exactly is that?
What are aphids?
Yeah.
Aphids are an insect pest. One of the bigger problems in the cannabis industry are aphids. Most folks control it by just having a really clean grow environment. But even then, aphids can still proliferate. So what they do is they latch onto the plant and then they suck the sap, sort of the moving sugar water through the plant. So it just leads to lower production from a plant standpoint, overall yields, cannabinoid content, terpene content.
So I want to change gears a little bit. You mentioned tissue culturing. Could you kind of provide a simple definition for our listeners of what tissue culturing is? Why is that different from traditional breeding methods where you're just kind of crossing 2 plants and letting them grow and kind of doing the Punnett square? What was the guy with the beans?
Gregor Mendel.
Yeah.
Green wrinkled and yellow round.
Yeah, exactly.
Your teacher would be so proud, Jordan.
Oh man. So actually tissue culture is not something that we use in our breeding schemes. At the very basic level, we do go back to Punnett squares. That's what we refer to as Mendelian genetics. So when we're capturing all this big data, this, genetic sequencing data, you can start to see traits segregate in what we call Mendelian fashion, where it's 1 to 2 to 1, which is exactly the point of doing those Punnett squares. So, but to get to your question on tissue culture is a way for clonal replication, meaning, right, in the cannabis industry, people are always taking clones of their plants. It's, cutting, you give it some rooting hormones, you put it in some rock wool or maybe some soil, and you get roots. Tissue culture is just a different way of doing that. And it's done on a much smaller scale from a plant perspective, right? For a healthy clone, or you want to take a 6-inch cutting I don't even know the number, but for micropropagation or tissue culture based cloning, you're taking something that's a few millimeters in length. And so it's just the same thing. You're giving a cutting of the plant nutrients that it cannot get without roots, and you're tricking it to grow roots is the goal. So that's tissue culture. In the cannabis industry, there's some companies that do their cloning methods that way rather than the traditional cloning. And it provides the benefit of young clone is grown in a completely sterile environment, which there is evidence out there that you can remove some, old accumulated systemic diseases or something from a tissue by using tissue culture. We've never done that. And I don't want to make the claim that we can do that because I have no idea on the legitimacy of those claims. But that's tissue culture as it relates to cannabis in the short.
Appreciate that.
Yeah.
Let's talk about Anchor. If you haven't heard about Anchor, it's the easiest way to make a podcast. Let me explain. It's free. That's right. No more excuses. Get your lazy ass off the couch. Go start a podcast. There's the creation tool that allows you to record and edit your podcast right from your phone or computer. Once again, no more excuses. Anchor will distribute your podcast for you so it can be heard on Spotify, Apple Podcasts, and many more. Couldn't be easier. Even better, you can make money from your podcast with no minimum listenership. That's right. They're paying us for this ad. Thank you very much, Anchor. Download the free Anchor app or go to anchor.fm to get started now.
I've got another side question for you. Can Dewey play a major role, or can it play a helping role in the future with the personalized medicine by understanding exactly how the whole process works? Is that somewhere you see Dewey playing a role in the future?
As it relates to personalized medicine, I think that, the combination of all the phytocannabinoids and the terpenes within the plant are consistent, right? If you have, and this ties into, it's the entourage effect, right? If someone is using cannabis for medical purposes, they want to get that same mixture every time, just like you do with, like aspirin. It's always going to be the same ingredients time and time again. So for folks going after a more natural means of pain relief or whatever reason, you want to find out what works for you. And then you want to make sure that those chemical constituents are always there. So where we can come in is, we will know that in a given environment, this strain will always produce this chemical profile. And so there's multiple factors there. It's the environment, it's the genetics, and it's what the end user is getting, which basically comes down to the processing of the flower or the extract, whatever it may be. So from a genetic standpoint, it's making sure that the plants that are being sent to the growers of, medical cannabis growers, or even the patient themselves, that the genetics that they're receiving are at least capable of providing the chemotype that treats their ailment. There are variables in it that are out of the genetics control, but for the most part, if the genetic makeup is there, the chemical profile will be there to help.
The environment is got to be such a complex variable to account for, and especially given all of the restrictions now with large MSOs kind of growing across different states. I mean, how does that work, Jordan? I mean, you got major companies across state lines all trying to grow roughly the same style products so that when you go to dispensary in Curaleaf in California versus in New Jersey, you can kind of feel familiar with the products, but there's got to be a ton of different variability. So how does that work?
Yeah. So from a genetic and biochemical perspective, like the major pathways that are producing medicinally relevant or even bioactive components, right? We're talking about the cannabinoids, we're talking about the terpenoids, and we're talking about the flavonoids. All of these pathways are heavily influenced at the genetic level in terms of genetic expression, which is really the driver, right? So if you think of back to something you learned in biology with Gregor Mendel, is the central dogma of biology, which is genes are at the first level. They get transcribed into RNA, which is the gene expression, and that gets translated into proteins. And the proteins are like the chemical reaction factories. So all of these pathways, cannabinoids, flavonoids, terpenoids, are incredibly sensitive to environmental interactions. And so in the genetic space, we call this gene by environment interactions, G by E. And so that is something that is, I think, the next big hurdle to clear in the cannabis space. Folks are getting really good with their genetics. The next hurdle, I think, to clear in producing that consistent product from state to state to state is ensuring that the environment is the same, so that these metabolic pathways that produce these bioactive compounds are operating the same. Now, from a recreational standpoint, maybe the chemical the chemotype isn't as important for the consumer. Maybe somebody just wants like super frosty buds, or they only want purples. Those are two traits that are absolutely controlled by the genetic makeup. But if we're talking about purple, you could have a purple plant when it flowers at 72 degrees, it's purple, and when you flower it at 82 degrees, it's mostly green. So that's an example of a G by E trait is plant color. And then something else like trichome preservation. That's like another big thing in the processing space right now. Making sure that your trichomes don't wither because you want to have big frosty nugs for the recreational consumer. That also, might lead to a more potent flower, but also you're retaining the terpenes better. So it doesn't matter how good genetics are if these other steps aren't taken care of.
Is there a way that you could potentially implement like genetics testing throughout the life cycle of a plant as like a quality control mechanism?
Yeah, it's a technique that's a little pricey at the moment, but it's referred to as RNA-seq. So RNA sequencing, and what that does is gives you an idea of how the genes are being expressed. And so that's actually a service that we do offer at Dewey with other folks, is we'll look at your plants at different times. And so we can see when terpene genes are at their highest. And then, we can modify the grow environment to try to then, okay, how can we maximize expression of these genes? And that's something we offer and have done at Dewey.
So say like I have my HVAC goes down one night, right? And I come in the next day and it is a completely different environment. I then call you up, you come in, we're able to determine kind of what's going on from a genetics perspective. Is that something that you could then coach us through on like how to correct the environment to try to get that RNA sequence back to where it should be? Or is that kind of like, well, you just accept it and move on kind of a situation?
Yeah, the results aren't going to be immediate, right? If your HVAC goes down, you got to get your HVAC back up and running, or your host, right?
Yeah, of course.
But let's say, right, you're a company in California and you're growing indoors, and you get a license for New Jersey, and you want to make sure your products are the same. Well, first off, your genetics need to be the same. But yeah, so let's say you start growing in New Jersey, your plant, the chemical profiles is not there. This strain that just smelled so good in California, it doesn't quite smell as good in New Jersey. We could answer that question with RNA-seq. We'd look at the gene expression profile of the same plant grown in what you think are identical conditions, and then diagnose what's wrong using that gene expression fingerprint.
Sounds like you're the Ghostbusters to me.
Yeah, that's the thing. A project like that, that's not an immediate timeline. That would probably be a 1 to 2 year project.
We're going to cut that part out from the 2 year timeline. So people just hear, this guy can come in, he can solve problems immediately.
Yeah, let's raise expectations, please.
Kellan, did you have a question you wanted to ask?
I was just going to, I thought you brought up a good point, because I have consumed, say, like an OG in Colorado, and then you go to California, and say I'm in Southern California and I purchase another OG, it's completely different, versus even up in Northern California, there's another OG, and it's like they're 3 completely different plants, but they could have all started. Do you think they all start with the same genetics, but based on the environment, that's going to change it? Or is it we're just at the point where people don't really know what genetics they're really starting with yet? Like, could you kind of walk me through why that is the reason from a genetics perspective?
Yeah, I think the first reason is it's probably not the same genetics. I think it's really unlikely that the clone from one source is going to be found all over the place. I mean, that's what the cannabis industry is it's ripe with copycats, which is fine. But when it comes to understanding that from genetics, there are tests out there that will answer that question. Medicinal genomics has a great product for determining the lineage of something. So, right, the first step would be, okay, are the 3 OGs in these different locations actually the same genetically? If no, well, there's your problem. If yes, then absolutely. Gene by environment is most likely driving that change.
Appreciate that.
Yeah.
Yes. My next question would be, what percentage of the industry is using kind of techniques similar to the ones Dewey provides? And is it based on the sheer size that's the determining factor whether they move forward or not?
What do you mean by size? The sheer size?
The size of the company, right? So like
size of the company.
We've got like boutique style growth who might be more geared to using it, and you've got like kind of larger ones where I'm curious to know, like from your standpoint, what percentage of the industry at this moment, both hemp and cannabis, is using in Dewey Scientific or similar style analysis?
Oh man, probably 1% or less. Really, the companies that are doing this are in the hemp space, the breeders, like the really good breeding companies. And in the rec space, yeah, there's a few folks that are doing stuff. I think mostly in the rec space, the companies that are doing this are in Canada. And we're talking, right, the giants up there. And because, for the size of the company, that's really our tools are more built for companies that are trying to get the same consistency in their products from different locations. So, right, like a small farmer in Washington that is craft premium flower probably won't have much benefit from working with us. But the folks that are trying to replicate their product, provide consistency to their brand, those are the folks. Their brand in multiple locations are the folks that would benefit most from what we offer.
Seems like a no-brainer for the large MSOs, especially the ones that are like opening up shop in new locations as fast as humanly possible. You need to have some sort of consistency, especially as people want that. So it seems like a no-brainer for them.
Yeah, I think to some degree it ties so much into consumer expectations of the brand. And because like some states don't have vertical integration where the retailer is actually growing it, it's something that's sort of hard to control. But for a multi-state operator that is plant touching only, it makes no sense because, right, they want their brand to be the same no matter where it is. Like Coca-Cola, the Coca-Cola at my Chevron down the street is the same as the Coca-Cola where you are. And so for these multi-state operators, you got to have these quality control pieces in place. And genetics is a huge component of that.
Is this technology that's kind of unique to the cannabis industry, or is this used in other big ag?
Yeah, it's definitely used in other big ag. I think a really good model is the berry industry. The largest berry provider in the US, at least, is Driscoll's. I'm sure you guys have Driscoll's in New York and Colorado. Their entire business model is built on their R&D program at the genetic level. So that's their first step of quality assurance, is they have kick-ass genetics. The next is they select their partner farms, and they set pretty tight rules on what the farms can do and how they grow their berries, and everything from the pesticide treatment to the fertilizers that are grown, right? So that comes down to the G by E, the gene by environment interaction. And then beyond that, they have, very strict and standardized processing procedures, right? Like everything that's A quality goes into their Driscoll's brand. Everything that's B quality goes into their other brand. Anything below that gets sold to somebody else, or they turn it into puree. So that's an example of a different industry using these same tools that we're talking about at Dewey.
Guys, I want to talk to you today about one of our new partners, CESC. CESC is a nonprofit organization providing a compelling and complementary alternative. They represent the ability to harness a flexible, collaborative approach to scientific advancements. They are comprised of leading doctors and researchers in the cannabis and cannabinoid science space for almost a decade. Their act first, talk later operating principle has now led to a successful series of disruptive innovations in the cannabis science space. They need your help now. Join them, collaborate with them, or support them. Go to thecesc.org to get involved now. Together, we can change the world.
What's the future roadmap for Dewey looks like?
Yeah, future roadmap right now, we are building our consumer brand. We are breeding, always breeding. And we're almost to the point where we're going to start licensing our varieties to folks in Washington. And right, these are varieties that are resistant to a myriad of pests, confirmed genetically, and then that compete with some of the popular varieties out there from a cannabinoid and terpene perspective.
Your biggest misconception since you started working in the cannabinoid space?
So from the time that I started working with cannabis to now, what was my biggest misconception? Oh, geez. I thought that we would have a larger barrier to entry than we did, right? Dewey was founded 3 years ago. I think at the time there was like 8 states that had legalized it recreationally, not many. And now we're in the 20s, close to the 20s. So I think that was just a misconception. I was like, oh man, no one's gonna be able to do this anywhere else except for, a handful of places. Now it's like, oh shit, it's everywhere. But before, starting Dewey, I'd worked in like medical collectives in California. I've been smoking weed a long time. So I shouldn't say there was any major misconception. I sort of knew what I was getting into. I think the passing of the 2018 Farm Bill with legalizing hemp threw me for a loop. I was like, hemp, who cares? Like, who's going to be using CBD? You're not getting high from hemp. So that threw us for a loop and made us question if we should do any sort of pivoting. And we did, of course, we started working with some hemp folks. But yeah, I would just say the growth of both sectors, hemp and adult use cannabis, I didn't think it was going to be this fast.
Yeah, the industry moves just incredibly fast. And it's funny, right? As we go on this journey where the states announce, they're coming online, there's huge progress. But at the same time, it's like incremental, and we're still in the first inning, which is like the craziest part. We're literally just beginning.
Yeah, exactly. I don't remember who said it, but a week in the cannabis industry is like a year in anything else. But at the same time, right? Like we don't have our act together on a federal level. Like we can't even, neighboring states can't trade product. We do still have a long ways to go despite the speed of the last few years.
We're still trying to figure out banking, which by the time this podcast is released, we'll have it figured out, is my hopeful assurance.
Oh man, I really hope so. Oh man, that was a big
in this podcast out until it goes down. No, it'll happen. We're going to get it done. I mean, you can see they're close, but it's crazy, right? Like we have people all the time that are outside industry that want to get in and are just perplexed by the type of obstacles that are kind of like self-imposed on an industry that is still fighting and thriving at the same time. And I think that's the most impressive part, is that some of these companies, including yourself, are doing really challenging next level things and working with partners that are thrown for all these additional obstacles.
Yeah, it's, you mentioned banking as a big obstacle. The other one is the tax situation. Like we have to, 280E. I don't know if you guys have discussed this on your show, but
no, I like it.
This tax code that says the only thing that a provider of a narcotic, which of course cannabis is still labeled as such, the only things you can deduct, right? You can break the law by trafficking narcotics, but you better not write off your accounting expenses. Like the only thing you can write off are the costs of the goods sold. So like for a cannabis grower, it's like the soil, the lights, the pots, the fertilizer, and the people that grew it, that like their salaries. So like that's something that when I talk to folks outside of the cannabis industry and they learn about that, they're like, are you kidding me? And it's just something that's ludicrous. And it goes hand in hand with banking. Like the financial perspective of how the cannabis operates right now, it's amazing that there are companies, like you said, Bryan, that are thriving the way they are. Some of these MSOs and all the hoops they have to jump through, like at Dewey, we have to deal with one governing board of the Washington State Cannabis and Liquor Board. And like, I can't imagine what like Curaleaf has to deal with. Like we dealing with one entity, one government regulator is enough. So props to them.
I think that's what they have, a team of lawyers. By a team, I mean like an entire baseball team full of lawyers, because it's got to be nearly impossible. And the 280, I don't want to dive into that because it's a whole nother topic on its own. But when that gets removed, these operators will just have a ton more profit. And people are like, whoa, these guys made a huge amount of money this year compared to last year. Like, what did they do different? And it's like, no, the government stopped screwing them over.
Yeah, that's a good investing tip, Bryan.
I got to have to do a disclaimer at the end of this podcast now. We are not professional investors. Do not take our advice. But yeah, wink, wink. Absolutely. Before we wrap, we're going to do 2 questions we ask all of our guests. You could sum up your experience in the cannabinoid space into one main takeaway or lesson learned to pass down to the next generation. What would that be?
Keep your end goal in sight. You don't know what's going to get in your way of getting there, but as long as you have your eyes on what you want to get in the cannabis world, I think you can achieve it.
The last time you consumed any cannabinoids?
Oh, before this podcast, I took my dog on a walk and smoked a joint with him.
Love it.
Yeah.
I was going to ask you guys if you guys wanted to have a little toke sesh before we started this. I'll get on the same level, but we just got into
We'll have to save that for the Patreon members for the video subscription.
There we go.
But yeah, absolutely. We should definitely get together. Now we'll have to do a virtual one, but for soon in person. So prediction time. Will the future be genetically engineered cannabis? If so, what does that look like from a time frame perspective? If not, why not?
That is a great question. I don't know if it's tough because like cannabis is an industry that has its own subculture that has thrived within America, right? And it's often associated with preserving nature, right? It stems from the hippie movement, the free love movement. And so for those types of consumers, GMOs they're never going to consume GMO cannabis. So I think any company that is producing GMO cannabis is alienating sort of the base of cannabis users. Now, that's not to say it's not being used today by certain people. I just don't think that it's GMO cannabis will ever really hit shelves just because of the makeup of consumers. Now, using it in breeding programs as a tool to learn is probably the way that it's going to be used, and it's already being used by a few companies. And then that's in the plant aspect of cannabinoids. The other side is there are a bunch of companies right now that are either currently producing or on the verge of commercially producing engineered yeast grown cannabinoids, where they've genetically engineered yeast to have the entire cannabinoid pathway, including the precursors. So they're feeding these yeast sugar, and they're in turn producing cannabinoids. That's I think going to be a very useful tool as it relates to cannabinoids as a medicine, and is going to be key for production of what call it the rare cannabinoids, or even cannabinoids that don't really exist in nature but might have clinical relevance for a certain disease or ailment.
Kellan, thoughts?
I mean, metabolic engineering is really close to my heart. It's what I studied for a long time. And so I agree with everything that Jordan said. I don't think that, GMOs aren't even allowed in Europe right now. Like there's no way the demographic of cannabis users are going to entertain the thought of consuming a genetically modified cannabis plant. So, but as far as the genetically modified organisms go, like yeast and E. coli, I do think that is just going to continue to gain traction. But I do think at the end of the day, it's only going to be for pharma, because it goes back to the thing. If you are producing, say, a THC molecule from E. coli and you're using it to make a vape pen, that vape pen is going to have to say it's from a genetically modified organism, and no one's going to buy it at that point. So I think it's going to be strictly regulated to kind of the pharmaceutical land. Brian, what do you think? What are your thoughts here? Would you consume some genetically modified cannabis?
If Jordan was behind it, absolutely. But at the end of the day, I trust Jordan blindly. So the average consumer is not going to be like, there's too many things going on, guys. Like they're not going to evaluate a product. At least I don't think they will. I don't think they're going to pick up a product, read the back. Like I don't do that now when I buy products. So like I can't imagine when you go to a dispensary, you buy these products. I don't think that people are going to be as informative in that. And I think you'll have your slice of individuals who are really well read and understand exactly what they want to consume, like you do in other industries. And I think for the most part, it's all about money, right? At the end of the day, what makes the most sense? What's the cheapest way to mass produce these things from a clean standpoint to get past regulations? And then go from there. So I'm going to take the other side.
I like that. One of us will be right.
Well stated.
For sure. Jordan, before we wrap, where can our listeners get in touch with you if they want to learn more?
If you want to learn more, check us out at DeweyScientific.com. If you're interested in our recreational cannabis brand, check us out on Instagram. We're at Dewey Cannabis. And then, of course, DeweyCannabis.com for our recreational features.
We'll tag them all in the show notes so people can find you guys. Thanks, Jordan, so much for your time. And thank you, everyone, for listening.
All right. Thanks, Brian. Thanks, Kellan. Had a blast.